All right, today we're diving deep into the world of antigen-antibody reactions. Now, this might sound a little technical, but trust me, this is the invisible engine that powers some of the most important diagnostic tests we have in medicine. We're going to break down exactly how it all works.
I mean, think about it for a second. How is it possible that a lab can take one tiny drop of your blood, which is just swarming with billions of different molecules, and find the one specific marker for a single virus? It seems like magic, right? Well, it's not magic. It's an incredibly powerful and elegant biological partnership.
So here's our game plan. We'll start with the fundamental bond itself. Then we'll get into how we measure its strength, think strength versus synergy. After that, we'll hit the Goldilocks zone, which is crucial. And finally, we'll walk through both the classic and the super modern lab tests that make all of this stuff visible and, well, life-saving.
Okay, first things first. We have to understand how these two molecules, the antigen, which you can think of as our target, and the antibody, our seeker, how they actually connect in the first place.
The first thing to get is that this isn't some permanent chemical reaction. It's much more like a very, very specific handshake. The bond is strong, but it's also reversible. Nothing gets permanently changed, and it's held together not by one single massive force, but by a whole bunch of weaker ones working together.
And these are the four forces right here. Now, individually, they're pretty weak. But here's the kicker. For an antibody to bind to its target antigen, they have to fit together almost perfectly, like a key in a lock. And that closeness allows thousands of these weak forces to all grab on at the same time, creating one seriously powerful collective bond.
The best way to picture this is like Velcro. Seriously, if you just have one little plastic hook and one little loop, you can pull them apart with no effort at all. But when you have thousands of them lined up perfectly, suddenly you've got an incredibly strong connection. That's exactly the secret behind the antigen-antibody bond.
So now we know how they stick together. But how do we measure the strength of that connection? This brings us to two really important terms you need to know: affinity and avidity.
This slide really gets to the heart of it. Affinity is the strength of just one connection, one little binding site on the antibody grabbing onto one specific spot on the antigen. That spot's called an epitope. But avidity, that's the total strength. You see, antibodies have multiple binding sites, multiple arms. Avidity is the combined strength of all those arms holding on at once. And it's way, way stronger than just adding up the individual affinities. It's pure synergy.
Okay, so we've got these specific, strong bonds. But this is all happening at a molecular level. It's completely invisible. So how in the world do we actually see a reaction in a lab test? To do that, you need millions of these pairs to work together. And that means getting the proportions just right.
And this is the brilliant insight from Merrick's lattice hypothesis. For a test result to actually become visible, the antibodies, which have at least two binding arms, have to grab onto multiple antigens at once. This cross-linking action builds a massive, visible web, or lattice. But, and this is a big but, this only happens under very specific conditions.
So let's look at what happens when it goes wrong. In what we call the prozone, you have way, way too many antibodies. Every single antigen gets immediately swarmed and covered, usually by just one antibody each. There are no open spots left for other antibodies to create those crucial crosslinks. The result? No lattice, no visible reaction, and a potential false negative. And then you hit the sweet spot, the zone of equivalence. This is the Goldilocks zone where the ratio of antigens to antibodies is perfect. Each antibody can grab multiple antigens, and each antigen can be grabbed by multiple antibodies. This creates that huge, stable, interlocking network that finally becomes visible to the naked eye as clumping or cloudiness. This is what we're looking for.
And on the flip side, you have the pozone. This is the exact opposite problem. Now you have a massive excess of antigen. Every single arm of every antibody gets completely saturated by a different antigen. Once again, there's no way to form those bridges between antigens, so no lattice can form. And again, this leads to a false negative. It just goes to show you getting that ratio right is absolutely everything.
So how do we actually put this Goldilocks zone principle to use in the real world? Well, let's take a look at some of the foundational tests in immunology that are built entirely on creating that visible lattice.
The two main types you'll see are precipitation and agglutination. And honestly, the only real difference between them is the state of the antigen. In a precipitation reaction, the antigen is soluble. It's dissolved in a liquid. When that lattice forms, it gets heavy and falls out of the solution as a visible solid, a precipitate. In agglutination, the antigen is already attached to a big particle, like a red blood cell.
So here, the lattice forming just causes all those big particles to clump together.
A really classic example of a precipitation reaction is the VDRL test. For decades, it was a primary screening tool for syphilis. It's basically looking to see if antibodies in a patient's sample can form a visible precipitate with a soluble antigen right there on a glass slide. Simple, but effective.
And for agglutination, the most common example, one we've all heard of, is blood typing. It's the perfect showcase. When you mix a blood sample with antibodies against A or B antigens, you're just looking for that clumping. If it clumps up when you add anti-A antibodies, you know for sure that A antigens are on those red blood cells. It's a powerful, visual, direct application of the lattice hypothesis.
Now, those classic tests are great, but sometimes they just aren't sensitive enough. So modern diagnostics took things to the next level. What if, instead of waiting around for a visible clump to form, we could just tag our antibodies with something we can easily measure, like an enzyme that triggers a color change?
The indirect ELISA is designed to hunt for antibodies in a patient's blood. Here's how it works. First, a lab plate is coated with the antigen. Let's say a piece of the HIV virus. Then you add the patient's serum. If they have HIV antibodies, they'll grab on. Then you add a second antibody that's been tagged with an enzyme. This one is engineered to stick only to human antibodies. Finally, you add a chemical substrate. If that enzyme tag is there, the well changes color, and boom, you've confirmed the patient's antibodies are present.
The sandwich ELISA totally flips this around. It looks directly for the intruder, the antigen itself. First, the plate is coated with a capture antibody. Then the patient's sample goes in. If the antigen, like rotavirus, is in there, it gets caught. Next, a second enzyme-tagged antibody is added, and it binds to a different spot on the antigen. You've literally made an antibody-antigen-antibody sandwich. You add the substrate, and the amount of color tells you exactly how much virus is there.
Okay, the competitive ELISA is a very clever twist, but the logic is kind of backwards, so follow closely. In this test, you add the patient's antibodies and a set amount of enzyme-tagged antibodies all at the same time. They have to compete for a limited number of binding spots on the antigen. So this means the result is inverted. The more antibody a patient has, the less color you see, because their antibodies outcompeted the tagged ones. A lack of color actually means a positive result.
Now, when a screening test like an ELISA comes back positive for something as serious as HIV, you need absolute certainty. You need the final word. That's where the Western blot comes in. What it does is first separate all the individual proteins of the virus by size, creating distinct bands. Then we see exactly which of those protein bands the patient's antibodies are actually reacting with. It's not just a yes or no anymore. It's a highly specific fingerprint that confirms the infection beyond any doubt.
And the technology just keeps getting cooler. Techniques like immunofluorescence use fluorescent dyes to make these antibody reactions literally glow under a microscope. And then you have immunoelectron microscopy, which is so incredibly powerful, it lets us see the actual clumps of virus particles being held together by antibodies. It's just amazing.
So when you really think about it, from a simple blood type test all the way to a definitive HIV confirmation, this fundamental, microscopic dance between an antigen and an antibody is at the very heart of it all. It's an amazing testament to how understanding the most basic rules of biology lets us build tools that save millions of lives. It really makes you wonder, what will the next breakthrough look like?